Hello everyone. In today's class, we are going to have a comprehensive overview about the chapter Plant Growth and Development from NEET perspective. So the first topic here is plant growth and development. Now the PYQs perspective, if we see definition of development and growth, difference between the both is important. Life cycle of the plant, fundamental points about the plant growth, what are intrinsic factor, what are extrinsic factor, and the key questions which we have in the NCERT book are important. So when I talk about development, that includes when a plant is it starts from a zygote, it becomes a seed, it is then a seedling, then plantlet, mature plant, senescence, and death. So whatever different phases a plant undergoes in its life cycle, that is called as development in plants. Now this development is the sum of growth and differentiation. What is growth? Growth is equal to the increase in the protoplasm of a cell. And differentiation, a cell when it acquires functional specificity, the tracheids, like a meristematic cell, when it forms a trachid and it performs a special function of absorbing water, that is called as differentiation. So development is equal to growth plus differentiation. Okay, then we have some key questions here. That is, first one is, why does vegetative phase precede flowering? Okay, now vegetative phase it precedes the flowering so that plants have a strong structure. Okay, and like it has the essential structure of roots, stems, leaves, and they have enough energy so that it can support the energy intensive process of reproduction. Okay, that's why vegetative phase precedes the flowering. Next question is, why plants grow indefinitely but leaves or fruits are limited? Because the plants have certain tissues which are meristematic tissue, and they have the capacity of indefinite or indeterminate growth, while the leaves and fruits, they are determinate organs. That's why they do not grow infinitely. Okay, next question is, why cells from the same zygote show different structure? This is because the same zygote can undergo the process of differentiation, and it can show different structures or different functional specificity. Clear? Then the important fundamental concepts are, all the plant cells that descend from the zygote. The growth is very ordered and sequential, and it depends upon intrinsic factor, intrinsic or internal factors, which includes genetic or the genes and the plant growth regulators. Then we have external or extrinsic factors. Examples are light, temperature, water, oxygen, nutrition, and gravity. Okay, so let's look into the next topic. Now if you see the second topic, that is growth types, phases, and measurement, mostly questions are asked from the definition of growth, then it's why growth is called as indeterminate or open, indeterminate and open. Then the different type of meristems, that's another favorite, like you have primary meristem and secondary meristem. Then characteristic of the meristematic phase, elongation phase, that is also very important from exam perspective. Then if you come to measurement of growth, then how is like pollen tube measured with respect to length, and how is leaf measured with respect to surface area? This is also another point. Then some data questions also come, like maize root, how much one maize root cell it divides per hour, and how much a watermelon cell increases by size. So that is another important area. Okay, so these are the important areas from questions come. Let's look the concept now. So what is growth? Growth is equal to the irreversible or permanent increase in size, mass or number. The term irreversible and permanent increase is very important. Okay, now growth is called as indeterminate in case of plants because it is unlimited in nature. It can grow continuously. Why? Because it has special cells called meristematic cells in them, capable of continuous division. It is also called as open because new cells are added always. This meristematic cells, it can be of two types: primary meristem, which is present responsible for primary growth. Example is root apical meristem and shoot apical meristem. Root apical meristem adds the cells in an upward direction. Shoot apical meristem adds the cells in the downward direction, right? Secondary meristem is responsible for increasing the girth, and example is vascular and cork cambium. Okay, what is the primary meristem function? It increases the length, okay, or helps in elongation. Next, come to measurement of the growth. Growth can be measured with respect to increase in length in the case of pollen tube. If you consider leaf, then increase in area. Other parameters are volume, increase in fresh or the dry weight, increase in the cell number. Then we have some two most important examples, that is a cell of the maize root. It divides 17,500 cells per hour. So the term is very important here. And a watermelon cell, it increases in size by 350,000 times. Next, come to phases of growth. So if you remember this diagram, in the bottom part we have the meristematic phase, which is characterized by thin walls, dense cytoplasm and big nucleus. These characteristics are important. Thin walls because it helps in division, and big nucleus because dividing cells are big nucleus. Dense cytoplasm, actively dividing, needs more nutrients, so dense cytoplasm. Then you have proximal to the meristematic phase, you have elongation phase, which is characterized by vacuolation, increased vacuolation and cell enlargement. Increased vacuolation, why? Because it needs more water. More water means more vacuole. Then we have maturation phase. Not much increase in the cell size. Okay, cell walls are thickened and specialized. They carry out specialized function. Okay, so this is about this topic. Then let's come to the next topic, that is growth rate and curve. So from this topic we can expect both numerical question and concept based question. So definition of growth rate concept, conceptual question comes. Then again arithmetic growth rate, what is linear growth, what is the arithmetic growth rate and what is the geometric growth rate. From there also question comes. Then the formula for both the types of growth is also important, right? From there numerical questions may come. Then another important term is what is absolute growth and what is relative growth. Like formula wise question can also come from these two topics. Then like growth rate comparison, like that typical example of two leaves, one bigger and one smaller, like relative growth rate, that is also very important. Then we have next topic is conditions required for growth. So you should just know like what parameters we need and what do they help in. Okay, so here you should know what is growth rate. Growth rate is increase in number of cells per unit time. Types of growth: arithmetic growth in which only one cell divides, another cell doesn't divide or differentiates, because of which we get a linear curve. So formula is LT is equal to L naught plus RT. This is your L naught value, that is initial length. R is the rate of growth. Okay, then so PYQs generally come from the identification of the curve and also from the formula. Okay, then you have geometric growth. Geometric growth in which both the cells divide. So we get a sigmoid curve here. So it will have lag phase, log or exponential phase and stationary phase. So it is given by the formula WT is equal to W naught E RT, exponential RT. Here R is the relative growth rate, or another term for it, efficiency index. It is very important. So in with they can give efficiency index instead of the relative growth rate and ask the numericals based on it. And another point, why in the stationary phase, why the growth is declined? It is because the nutrients get depleted. Okay, then you have absolute growth versus relative growth. Absolute growth is total growth per time. Okay, relative growth is growth per unit initial size. Okay, remember you have the two leaves, like absolute growth, if you see for both, how much increase in the surface area per time. Now if you come to relative growth, it means increase in surface area per initial surface area. That's how you get the relative rate. If you compare the both, then smaller leaf will have more or higher relative growth rate. Okay, this is very very important. Okay, then the like parameters required for growth, we have water, which is helpful because it maintains a turgidity and is the main medium of the cellular reaction. Oxygen, which helps in respiration. Nutrients are essential because it forms a protoplasm. Optimum temperature is required, and also light and gravity is required. Now here there is a PYQ trap. You pay attention here. Swelling of wood, it is not growth, it is by the process of imbibition. Okay, it's a physical process. Next topic is differentiation, plasticity and development. You can expect assertion based PYQs from this topic. It's a very important topic, and from here, from differentiation, dedifferentiation, redifferentiation, what is the definition of it and examples of it, that is important. And then open differentiation, that is differentiation is open, example with respect to tissue culture, that is important. And then we have development and plasticity. Here plasticity is very important. Now from plasticity, both the types, developmental as well as your environmental plasticity or heterophylly, in particular examples of it are often asked. Okay, that's the hotspot of PYQs. Let's read the context now. So differentiation is like when the meristematic cells perform specific functions. They convert into cells that perform specific function. Example, trachary elements which lose protoplasm. Okay, that is an example of your differentiation. Now dedifferentiation, what's the meaning of this? That the mature cells which have gained specificity, when they lose the specificity and become unspecialized, that is called as dedifferentiation. Example, interfascicular cambium and core cambium. Look here, this is a cross section of the stem, and blue is your phloem, red is the xylem. In between there is interfascicular intrafascicular cambium, and in between the bundles there is interfascicular cambium. So parenchymatous cells which are already differentiated under specific condition, they can dedifferentiate and form your interfascicular cambium or core cambium, which is example of dedifferentiation. Okay, what is redifferentiation? Excuse me, redifferentiation is nothing but, again, the cells which have dedifferentiated, they gain or they convert into cells that perform specific function. What are the examples? This interfascicular cambium, if it forms the secondary phloem and xylem, that's the example of redifferentiation. In contrast to tissue culture also, when the callus which is obtained from a section of a leaf or a node, like that, the differentiated cells, they again form root or shoot, then that is the example of redifferentiation. Okay, see, for tissue culture you have a leaf sample which is what, differentiated, it forms callus, that is a mass of undifferentiated cell. Okay, that is dedifferentiation. Then again that callus form root or shoot, that is termed as redifferentiation. Is that clear? Now let's come back to plasticity. So plasticity is the ability to change change with respect to the environment, or to the with respect to the developmental process. So example of it is heterophylly. Now we have two types of heterophylly. uh, that is developmental heterophylly, which is genetic in nature. That is, look, in the juvenile form the leaf will have one particular phenotype, and when it grows old, or in later stages, it will have another phenotype. Example of environmental heterophylly or genetic heterophylly, it is cotton coriander larkspur, CCL. Okay, very very important. Then we have environmental heterophylly. What is that? It means with respect to the environment, the plant will have different type of phenotypic structures. Example of it is buttercup. Okay, in the water, if you see, you have thinly sliced segmental leaves, and in the terrestrial leaves, if you see, it is broad in nature. Okay, so let's start with the final topic, that is plant growth regulator, which is the topic having highest weightage in PYQs. So some questions come from characteristic of plant growth regulator, that is small, simple, okay, low concentration active, and they act as inhibitor, abscisic acid and ethylene, promoter, Auxin, cytokinin, gibberellin, both ethylene, which is having more promoter activity than inhibitor activity. Okay, then we have roles, physiological roles of Auxin, gibberellin, cytokinin, ethylene and abscisic acid, which we have to pay attention. Okay, let's now see in detail here. Coming to discovery part, Auxin, it was discovered by Darwin, and Darwin in canary grass. Okay, but isolation was done by FW Went in where Avena sativa or oat plant. Okay, now bending of the coleoptile is due to formation of Auxin, which transfers to the shady part. Now for Wendt's experiment, agar block will have all the active ingredients, so it shows bending even though the coleoptile is removed. Then discovery of cytokinin was done by Scooch in tobacco internodal segment. Isolation, it was done by Miller in herringfish. First isolated cytokinin was kinetin, but it was from an animal source. First cytokinin isolated from plant is zeatin from zea maize or maize plant. Gibberellin discovery, it was discovered by Kurosawa from the rice plants suffering from Bakane disease or foolish seedling disease, where rice plants showed abnormal elongation caused by Gibberella fujikori. Isolation was done by Yabuta and Sumuki. Ethylene isolation was done by Cousins. Abscisic acid, three scientists named the same thing, inhibin B, abscisic, dormin. All three refer to ABA. Coming to chemical nature, Auxin is indole derivative, which is tryptophan precursor. Cytokinin adenine derivative. Gibberellin is terpene derivative, and ethylene is the only gaseous hormone, but commercially ethifone, which is a liquid form, is used. Okay, and carotenoid derivative for abscisic acid. Bioassay important. Auxin Avena curvature test, okay, in oats or Avena sativa. Chlorophyll test for cytokinin. Alpha amylase test or dwarf maize plant test for gibberellin. Triple response test for ethylene, very very important, and stomata closer test also very important for abscisic acid. Let's see specific physiological role of each hormone. Auxin, it promotes xylem formation, acts as weedicide. Example 2,4-D, 2,4,5-T. Now specifically for broadleaf dicots. Also rooting of callus is because of Auxin. Gibberellin, it causes bolting in rosette plants, increases the internodal distance. Example beet, cabbage. Gibberellin increases stem length and improves the yield in sugarcane. Increase in grape stock, improvement and size and appearance of apple is also because of it, and it is used in malting industry because it increases the alpha amylase activity. Come to cytokinin, it promotes the new leaf formation and nutrient mobilization. It increases the chlorophyll content. C cytokinin, C says chlorophyll. Okay, come to ethylene, it helps in fruit ripening, increases the respiration in fruit, very important term called as respiratory climactic. Here it improves the internode and petiole elongation in submerged rice plants. Okay, general internodal length increase gibberellin, but submerged rice plant ethylene. Abscisic acid, general growth inhibitor, prevents seed germination, causes stomatal closure, inhibits plant metabolism, induces stress resistance and drought resistance. Come to apical dominance, promoted by auxin, that's why used in pruning and hedge formation, and broken by cytokinin. So auxin and cytokinin are antagonists to each other. Cytokinin promotes lateral shoot formation. Seed dormancy, it is broken by gibberellin and induced by abscisic acid. But in peanuts, if you see, seed dormancy is broken by ethylene. Okay, fruit and leaf abscission, prevented by auxin and gibberellin, but it is promoted by abscisic acid and also ethylene. Ethylene promotes abscission of your cherry, and walnut also causes thinning of cotton. Very, very important. Senescence, if you see, it is prevented by auxin, gibberellin and cytokinin. Specifically, cytokinin is important. It is induced by both ethylene and abscisic acid. Okay, coming to cell division and elongation, it is promoted by auxin and cytokinin. Okay, auxin induces the root formation, cytokinin shoot formation of the callus. Flowering, if you see, in a female flower formation in by auxin, but specifically cucumbers, if you see, ethylene promotes the formation of the female flower. Okay, let's come to fruits. Parthenocarpy is induced by auxin, example in grapes and cucumber. And if you see fruit set in pineapple, it is because of ethylene, and fruit ripening in tomatoes and apples is also because of ethylene. So with this we completed the plant growth. With this, we completed the plant growth regulators. We covered each point in very short, but we covered all the important points, and also we saw them from PYQ perspective. So happy learning, everybody. If you like this video, please like, share and subscribe to my channel. Thank you, everyone. Have a very good day.